Thank you, Dr. Patel, for that introduction. I'm Dr. Emily Wearne, a pharmacologist and acting non-clinical team leader in the Division of Hematology, Oncology, Toxicology, or DHOT, at the FDA. Today I'll be giving an overview of CDER non-clinical resources and relevant guidances that are key to approaching first-in-human studies in oncology. Here's an overview of today's webinar. We'll discuss where to start, what we are looking for at the FDA, any potential pitfalls you may encounter, and general recommendations for your consideration. In general, non-clinical data includes pharmacology or mechanism of action data, pharmacokinetic data, and toxicology data. The role of a non-clinical reviewer at the FDA is typically front-loaded, such that we review non-clinical data submitted under new INDs to determine an initial safe starting dose for a first-in-human clinical trial, as well as what toxicities to monitor for in the clinic. Importantly, we function as part of a multidisciplinary team. So where to start? You've identified a promising target and shown that your drug affects that target. You've shown activity in an in vivo tumor model. But is that enough? And what's next? Well, we recommend that you start with the guidances. This slide contains titles and links for the non-clinical guidances that are most relevant to oncology development. Specifically in oncology, we use the ICHS-9 and the ICHS-9 question and answer guidances, which discuss non-clinical evaluation for anti-cancer pharmaceuticals, and this includes small molecules and biologics. Biologics are also discussed in ICHS-6. The ICHS-9 guidance explains the basic recommendations for anti-cancer drugs. These include 28-day GLP-compliant toxicology studies in 2 species, or a single study for biologics if you have scientifically justified that there is only one pharmacologically relevant species. These studies should use the intended clinical route of administration, and the dosing frequency should match or exceed that intended for clinical administration. However, deviations can be justified. In general, these GLP-compliant toxicology studies are the primary data used to determine the starting dose for first-in-human trials, although occasionally a general toxicology study is not conducted if there is no pharmacologically relevant species. And a useful tip here is that you can initially submit draft toxicology reports containing signed histopathology reports to the IND, and the final quality assured study reports would then be expected within 120 days of our receipt of the IND. ICHS-9 includes an exception on standard toxicology-based methods for determining the starting dose of immune agonists. It states, for biopharmaceuticals with immune agonist properties, selection of the start dose using a minimally anticipated biologic effect level, or MABEL, should be considered. A MABEL approach relies heavily on pharmacology studies. And in general, it's important to use the totality of data to justify the proposed first-in-human dose. DHOT has numerous publications evaluating methods of first-in-human starting dose selection for different types of products, and these have resulted from internal research projects. These publications are shown on this slide for your reference. In terms of toxicology study design, refer to the CIFSAN Red Book for detailed descriptions of toxicological endpoints for a large array of study types. So key endpoints in GLP toxicology studies are shown on this slide. And additional endpoints can be included if warranted based on your drug's mechanism of action. So for example, cytokines could be measured as needed for immune activating products. Good laboratory practice, or GLP, is a key concept in non-clinical development. GLP doesn't just refer to a study with good controls and endpoints. It also includes details about archiving, study conduct, and responsibility. GLP is described in 21 CFR Part 58 in the Code of Federal Regulations. Prior to initiating GLP studies, consider the contract research organization or lab that you want to conduct your studies, and make sure they can format the data in SEND, which is now required by the FDA for multiple study types. In addition, we encourage you to plan ahead. These GLP studies can be expensive, but they really are foundational for drug development. If you have questions or uncertainties about GLP study design, consult an expert first. You can also request a pre-IND meeting with the FDA before initiating the study. We can provide general feedback on major red flags and missing endpoints, but we do not design the study for you. So what else is recommended for a first-in-human study? Well, it depends on your product. According to the 1995 Content and Format of INDs guidance, INDs should contain proof of concept data, including a description of the pharmacologic effects and mechanism of action of the drug if known. We also recommend providing screening data and looking for primary and secondary targets, concentration response data, and in vitro cytokine release data for immune agonists. Safety pharmacology data, including CNS, respiratory, and cardiovascular assessments, are also recommended, and in vivo safety pharmacology endpoints are generally built into toxicology studies for oncology drugs. I'll now discuss some stumbling blocks and pitfalls that sponsors may encounter when submitting a first-in-human IND. According to a 2020 FDA publication, the most common non-clinical reasons for putting a new commercial IND on hold in oncology are concerns with the first-in-human starting dose and/or dose escalation, missing studies or supporting information, and severe toxicity in toxicology studies. Speaking of starting doses, we expect sponsors to scale by body surface area for small molecules and antibody drug conjugates, and by body weight for large biologics. If the drug is an immune activator, we expect pharmacology data to be submitted to support the selection of an appropriate starting dose. Potential pitfalls can also be GLP related. For example, your GLP toxicology study reports need to contain a signed pathology report. Only signed pathology reports are considered raw data. In addition, it's important to provide data supporting your selection of pharmacologically relevant species for toxicology studies that are conducted with biologics and oligonucleotides. And again, if needed, don't hesitate to consult an expert. So for your reference, this slide describes special considerations and additional resources, including guidances specific to radiopharmaceuticals, novel excipients, and botanicals, as well as information pertaining to antibody drug conjugates. And finally, we encourage you to request a pre-IND meeting. We now have interact meetings, which can be requested to get FDA input on unique challenges or the use of novel alternative methods or NAMs. A traditional Type B pre-IND meeting can also be requested, with follow-up opportunities available, including written clarifying questions. We recommend that your meeting package includes enough data for us to answer your questions, but don't submit full study reports, as these are generally not reviewed at the pre-IND stage. In addition, although we will provide feedback on your proposed approach to determining the first-in-human starting dose, we will not agree to a specific starting dose at the pre-IND stage because we have not yet reviewed the full study reports. So in summary, there's a lot of non-clinical data needed to open an IND. Let ICHS-9 be your starting point. Invest in good GLP-compliant toxicology studies. seek out a regulatory consultant if needed, and request a pre-IND meeting. As a closing thought, we hope you use these tips to improve your non-clinical package for your next first-in-human IND submission for oncology indications. This concludes my presentation. I'll be back shortly to answer your questions. Now I'd like to pass it on to Dr. Caitlin Tidings.